And now, on to our final youth speaker of the day, Rishika Podarala. Rishika is an undergraduate student majoring in microbiology and cell sciences with an avid interest in biological and genetics research. Rishika has published research about COVID-19 and won regional and state science fair awards on plastic degradation research. She also engages in community service as a member of the Indian Red Cross and is a bioscience MVP, a pharmacy technician certified, and is bilingual. Some of the hobbies Rishika enjoys include swimming, Indian classical dance, and traveling. Today, she will be talking about her work towards a more environmentally friendly way of disposing plastics. And without further ado, I welcome Rishika to the stage.
Hello, everyone, and thank you, Dimple, so much for that amazing introduction. One moment, let me share my screen. So like I said, my name is Rishika Podarala, and I'm here to talk about my research and how I learned that research is about more than just the science. So starting off with my research journey, I started in the midst of the pandemic. And although this was a very challenging time, I chose to seize the opportunity and engage in many virtual research opportunities. This really kindled my spirit for research. So I joined my experimental science research class at school. And this was taught in a completely virtual setting and involved a lot of independent study. Here you can see me at school working in the lab, and I promise I'm smiling behind the mask. So to dive a little more deeper into my research and what I did, this was my question. Which isolated bacterial species extracted from waxworms is most effective at degrading polyethylene? Basically, what I was trying to find was when I extracted the gut microbe from these waxworms and isolated them on different plate media, which bacterial species that I isolated would be most effective at degrading plastic. So I started off by feeding half of my waxworms oats and honey as a control group and half of them plastic. I then extracted the gut bacteria and then isolated it on 4 different types of plate media. Now, I decided to isolate my gut bacteria on 4 different types of plate media because each plate media has different nutrients that promote or inhibit certain bacterial growth. So this was to ensure that I could extract and isolate as many possible bacterial species as I can to see which one would be most effective at degrading the plastic. So moving on to my actual results, here in this table, you can see that the average percentage of biodegradation was highest in the plastic-fed waxworms. In addition, when comparing the different media types, the average percentage of biodegradation was highest in the cell plate of PEA. And just for some visuals, here are my bacterial samples that I extracted, organized by their cell media type. So to conclude, I found 5 bacterial species that were able to effectively degrade the plastic. And there's so much more research to be done in this area, but I truly believe that these waxworms can help us find new ways to degrade plastic in our environment. So now let's talk about the lessons I learned beyond the science, beyond my results. So lesson number 1, failures are just as important as successes in the research project. Here you can see my first method. In my first method, I decided to inoculate the petri dish with bacteria and plastic at the same time, but this resulted in the bacteria not being able to effectively degrade the plastic. So I took this failure and modified my experimental design and created a new method where I inoculated the petri dish with the bacteria first and then introduced the plastic. This ensured that the bacteria was able to concentrate on the plastic and effectively degrade it. See how my failure in method 1 led to a success in method 2? This goes to show that don't be afraid of failures. They can often help you learn and grow. Lesson number 2, communication and collaboration. Communication and collaboration are key in the scientific community. Often, during my experimental science class, we would all be in a group talking to each other, talking about our ideas and our research, and giving each other feedback. It's very important to talk with your peers because they can give you ideas that you maybe didn't think of before or help you look at your research from a different perspectives. You can also play off of each other's strengths so that at the end of the day, you are all successful. Lesson number 3, mentorship from professors. Mentorship from professors is very important. For example, in my research plan, I had many gaps throughout. So I reached out to a local professor and decided to utilize their expertise. Since they're so well versed in their area of study, they were able to help me perfect my research plan. Mentors can also help you brainstorm new experimental design procedures. For my research, I was having a tough time on coming up with a method of how to effectively find the average percentage of biodegradation of the plastic. So I collaborated with my mentor and we were able to come up with this new and innovative experimental design that led me to have very successful results. And last but not least, mentors can help you cross-check your research plan and give you constructive feedback so that you can have reliable and accurate results. Lesson number 4, perseverance is key. Remember, when you take up a research project, you are committing yourself to a lot of hard work and time. But trust me when I say it's all worth it. You'll make many mistakes along the way and you'll have to redo a lot of things. And that led me to spend hours after school and even come into school during holidays so that I could successfully finish my research. And that being said, I also made a lot of mistakes. Is a page from my logbook. You can see that I had to do 2 trials of the same procedure because I messed up in trial 1 and had to do it all over again to get accurate results in trial 2. But that's okay because doing this helped me learn and grow. And at the end of the day, doing both trials gave me a good foundation of comparison that helped me in my future research. Lesson number 5, simple solutions can solve challenging problems. Let me tell you again that along your research journey, you will face many unexpected hurdles, but don't let these stop you. For example, in my research, I had a solution filled with guts and bacteria. But the thing was, I only wanted the bacteria. So how was I going to separate the two? With just a simple syringe and syringe filter, I was able to extract the bacterial fluid, centrifuge it, and obtain only the bacterial pellet. See, using the syringe filter and syringe was such a simple solution, yet it led me to surpass this hurdle and move on with my research. Lesson number 6, the triangle theory. This theory was actually taught by my professor, research professor, and I found it very useful. So we all start at point A. When we start our research, we have a plan, a direction, and an aim. But sometimes we just keep going and we are not getting the results that we want, and we keep meeting with dead ends. But don't give up. Think about different perspectives of your research, and I guarantee you, you will find a great idea. So again, don't give up and think about your research from different perspectives. I'm sure you'll find a new idea. So that concludes my presentation. I just wanted to say a huge thank you to Youth STEM Matters for giving me the opportunity and platform to speak with you all today, and thank you all for taking the time to listen to me. Just one last note, a small call to action. Let youth motivate youth. See, we are the future generation of tomorrow. So let us talk to each other, collaborate, and support each other in our scientific discoveries so that we can have a real impact on tomorrow. Thank you so much.
Wow, Rishika, thank you so much for giving us all of this inspiration and calling us to action. I think I speak for everyone when I thank you not only for working to find solutions to these global problems, but also sharing with us all of these valuable lessons that you learned like throughout your research. And everybody, please be sure to share your own appreciation in the chat for all of these incredible lessons. Another reminder that you will find the last quest hint in the chat. To participate in the drawing, you will need to put all 6 phases together to make a sentence and submit your sentence via Google Form by 17:15 BST. So that's in about 30 minutes. You'll find the Google Form in the attendee pack, and previous hints can be found in the Research Conference Quest Expo booth. And thank you again,
so
Rishika.
much. And if anyone has any questions, I'll be sure to be in the chat to answer them. Thank you all so much again.